Okay, let's start chapter 13, the brain and spinal and cranial nerves. So first we talk about major landmarks in the brain, and we talk about the cerebrum. And there's two cerebral hemispheres. And there's a longitudinal fissure between these two cerebral hemispheres. Then in the very back of the brain, there is the cerebellum. There's the corpus callosum, which are fibers that connect the two cerebral hemispheres. And then the brainstem. It consists of three parts: the midbrain, the pons, and the medulla oblongata. So here's a side view of the brain, and the cerebral hemisphere here, the cerebellum here, and the brainstem here with the pons, medulla oblongata, and the midbrain you can't see. The evolutionary history of the brain is fascinating, and the brain is one of the most remarkable things there is. It's really something, but its design reflects its evolutionary history. And I make it like this. It's like if you're going to buy a 2021 car, a brand new car with all the bells and whistles and all the fancy stuff in a very modern car, but you have to put it on a Model T chassis, which is the very first car ever mass produced. That's how the brain is set up, because down deep in the brain we have a reptile brain still present. That old reptile brain is still present. Reptiles have the same parts of their brain that we have, but their cerebrum is very much less sophisticated. It's not convoluted. But that deep part of the reptile brain in our cerebrum, what's called the limbic system, and in our brainstem, it's still there. So there are paleo-mammal and neo-mammal innovations. And really, the innovation is the expansion of the cerebrum and the cerebral cortex, and becoming very, very sophisticated. But those old reptile and even amphibian pathways are still present, and we'll show you that a little bit later on, and how part of our cerebrum deals with strong emotions in a socially acceptable way, the way reptiles don't. They don't do things in socially acceptable ways, but we do. Okay. Embryology, we're not going to go into embryology of the brain very much, but remember in the tissue chapter, I think we talked a little bit about the three germ layers. An ectoderm becomes nervous system and is a neural tube that's formed under the influence of the nodal cord, and this happens very early in pregnancy, and so that's why we talk about neural tube defects and folic acid. Neural tube defects are defects in completing a neural tube, and one of them is called spina bifida, where the spinal cord is not completed and it sticks out from behind the baby, and this baby is usually a paraplegic. And the worst of the worst is called anencephaly, which is a baby that's born without a cerebrum. These can be prevented by taking enough folic acid. It's a B vitamin from green leafy vegetables, and it's critically important for forming this neural tube. It happens very early in pregnancy. So most physicians will advise young women, if you don't eat a lot of green leafy vegetables, you want to take folic acid. If there's any chance whatsoever you're going to become pregnant, you should take folic acid supplements. They're really cheap. You can take large amounts of them, and they don't really hurt you. And they guard against these neural tube defects, because these things happen very early in pregnancy, even before a woman really might even realize that she's pregnant. There's already a neural tube forming. Okay. Okay, gray and white matter. So gray matter is the cortex, cell bodies, and dendrites of neurons. White matter are the axons. And the axons in the PNS are called nerves, or bundles of axons in the PNS are called nerves. Bundles of axons in the CNS are called tracts. And then we're going to learn that the gray matter in the brain is on the outside, the white matter is on the inside. Well, there's some gray matter on the inside, but in the cerebrum, mostly the gray matter is on the outside, white matter at the inside. When we get to the spinal cord, we'll see that's completely reversed. White matter on the outside, gray matter on the inside. And then we can talk about the meninges, the coverings of the brain, the dura mater, or the strong mother. And with these dural sinuses in between the two layers of the dura mater. A sinus, remember, is a cardiovascular structure where you have lots of veins coming into a common area, and then one vein where the blood leaves that common area. And then there's the arachnoid matter, just underneath that's called the arachnoid matter. And that's the spider mother, because it has a space and it's got all these thin little filaments that look like spider webs in between there. And it's filled with cerebrospinal fluid. Then there's the pia mater, or the gentle mother. And that's the layer of a really simple squamous epithelium that lies right over the surface of the brain. Okay, here's a cross-section of the brain here, and here's gray matter, the cerebral cortex right here, and then there's internal gray matter called the cerebral nuclei that we'll talk about later on. And then here's the white matter in here, inner white matter, which are the axons from the neurons that are in the cerebral cortex. And here's the corpus callosum right here, where the two, these are called commissural fibers, where the two hemispheres send information back and forth and talk to each other. Right? And the internal capsule is more white matter, and those are more projection fibers. We'll get into these different kind of fibers later on, where the projection fibers are information leading from cerebral cortex neurons to lower areas of brain, and lower areas of brain send information up to the cerebral cortex. Then these ventricles, we're going to look at ventricles. There are spaces within your brain. Two lateral ventricles, and they're filled with cerebral spinal fluid. There's a third ventricle, and then there's a fourth ventricle. Okay, here's the cerebellum and brainstem cut in cross section. There's cerebellar cortex and white matter in this in the cerebellum. That's usually called the arbor vitae, but it's not listed up there. And there's the fourth ventricle right there. And then there's the medulla oblongata here. And then there's the spinal cord. As you see in the spinal cord, white matter on the outside, gray matter on the inside, completely reversed from what the brain is. Here are the meninges. So there's the skin of the scalp and a periosteum, where bone is covered by a layer of dense irregular connective tissue called the periosteum. Then there's the bone of the skull, and then there's a dura mater, the periosteal layer and the meningeal layer. And between those is a dural sinus. And in the dural sinuses are these arachnoid villi. See, underneath there is the arachnoid mater. And you see how all these spidery looking filaments in here. And they extend into the dural sinus on these arachnoid villi. And they do something important with regards to the circulation of cerebrospinal fluid. So we will talk about that a little bit later on also. And then the pia mater right here, just a very thin layer directly on the cerebral cortex. And these are all the meninges. Oh no, a falx cerebri is a dura mater that is between the hemispheres. In the lab, when we do the brain, we'll have to learn about these meninges in the sheep brain. So in the lab, we'll be looking at plastic human brain models, and we'll do actually look at actual brains, but sheep brains. And that's where you'll see the meninges, because the meninges, except for the pia mater, are not on the plastic models. Okay, ventricles and cerebrospinal fluid. So as I said, ventricle spaces in the brain are filled with cerebrospinal fluid. Two lateral ventricles, a third and fourth ventricles. So cerebrospinal fluid, it's a filtrated of blood. It's very similar to interstitial fluid. So it's really the interstitial fluid of the brain, produced by ependymal cells in the cord plexuses. Now we saw ependymal cells. We talked about those in the nervous tissue chapter. They are glial cells, and they make cerebrospinal fluid. And they're in the cord plexuses, and every ventricle has a cord plexus with ependymal cells making cerebrospinal fluid. Okay, so it's and here's the circulation. Circulated and reabsorbed by arachnoid villi in the dural sinuses. So I'm going to show you a slide later on about the circulation of cerebrospinal fluid. Sometimes called the lymphatic circulation. And I just learned something brand new about the importance of this lymphatic circulation. So cerebrospinal fluid is made in ependymal cells in the ventricles. It flows in the arachnoid space, in the arachnoid mater throughout the entire central nervous system, down the spinal cord too, and then is reabsorbed in the arachnoid villi into the dural sinus, into the blood of the dural sinus. Okay, functions of cerebrospinal fluid. Buoyancy for one thing. So your brain and spinal cord are floating in fluid, and they're neutrally buoyant. So they don't sink, they don't rise, they sit there in a cerebrospinal fluid. Protection. So your brain is the consistency of firm jello. So, and it's surrounded by bone. And if it hits against bone, it can be damaged. So one thing that helps prevent that is that if you hit, your brain's hitting against fluid, not against bone. Because if it hits hard enough, you will hit against the bone, and this is the essence of a concussion if you hit hard enough. Chemical stability. It has the correct concentration of all of the ions that you need for nerve transmission. Sodium, potassium, chloride, calcium, they're all in the correct concentrations. Chemical stability, so nerve transmission can occur. And then removal of toxic proteins. Here's the thing, your brain makes these toxic proteins. And one of the reasons for, one of the causes of Alzheimer's is the buildup of these toxic proteins, these plaque. And the circulation of the cerebrospinal fluid takes away these toxic proteins and puts them in the blood, and they get destroyed, probably by the liver. And this is one of the major functions of this lymphatic circulation. And here's the thing, it happens most often when you're asleep. Now it happens 24/7, but it really opens up, and apparently interstitial spaces become larger, and you really got a lot more of this flow of cerebrospinal fluid during the night. So this is a new, later on, we'll be talking about the functions of sleep, which are really mysterious. But one is not the removal of toxic proteins from your brain. And it's one of the major functions of sleep. And so here is a link between how much sleep you get and your risk for Alzheimer's disease. Who'd have thunk it? This is something I just learned myself just recently. Okay, and there is the circulation of the cerebrospinal fluid. It's all laid out, produced by cord plexus and the ventricles, by the ependymal cells there, flows from the lateral ventricles to the third ventricle, to the fourth ventricle, down the spinal cord, coats the whole outside of the spinal cord, comes back, covers the outside of the whole cerebrum in the arachnoid mater, and then it is reabsorbed across the arachnoid villi into the dural sinus. So it's filtered from the blood and then flows and then is returned to the blood. Right? Now there's a condition, which we okay, what's it called? It's called water on the brain, but it's not really water on the brain. It's a failure of the cerebrospinal fluid to be reabsorbed at the arachnoid villi, reabsorbed into the blood, and it continues to be made and it builds up. And let me look up its name. I forgot its name, so I'm gonna pause here and look it up. Okay, hydrocephalus. I should remember that. Water cephalus cerebrum. Oh, let me continue. Okay. And this causes, if it's not treated, this pressure of the cerebrospinal fluid pushes on the developing brain, and these little children get huge craniums filled with fluid and very tiny undeveloped brains, and it causes profound mental retardation. But it's easily treated. If it's diagnosed early on, they can put drainage tubes in and drain and artificially drain the cerebrospinal fluid. And then there's no problem. I had a student come up to me in one of the A&P classes when I was discussing it. He said, yeah, I was diagnosed with this when I was a kid. They put the drains in there and they're still in there. And you're gonna be in there your entire life to make sure that cerebrospinal fluid gets drained. Okay, barriers. We know about the blood-brain barrier, right? We talked about that in nerve tissue because that's the major cell responsible for that are the astrocytes. Remember, that's a glial cell. It's permeable to some things, but it's not permeable to a lot of stuff that other capillaries are permeable to. So they are the tightest capillaries in the body. And the brain really, the central nervous system is really a gated community. It's really particular about who lets in. And so, for example, for some drugs, like to treat Parkinson's disease, we'll talk about that later on, that's a lack of dopamine in what's called the substantia nigra. And so these people should be treated with L-DOPA, which is a precursor for dopamine, should be treated with dopamine, but dopamine can't cross the blood-brain barrier, so they modify this drug called L-DOPA. So it gets into the brain and it serves as dopamine. There's also a blood CSF junction. So there's tight junctions that prevent blood and CSF from merging. So cerebrospinal fluid is filtrated in the blood. But circumventricular organs, there are openings in this gated community. There are actually big openings because the hypothalamus has to monitor the blood levels of certain things. So they have to let blood in to the hypothalamus for this, and those are openings in the gate, and that's where sometimes big things and actually organisms and viruses can actually get into your brain if they find their way to these circumventricular organs in the hypothalamus. Okay, the cerebrum, as you mentioned, two hemispheres, left and right. A longitudinal fissure between them, and a corpus callosum is a fiber tract that allows, remember, the communication between the two hemispheres. And there they are, there's two hemispheres. And they divide up into lobes, frontal lobes in the front, obviously, parietal lobes in the side, occipital lobe in the back, and temporal lobes on the side, which you can't see there because this is a bird's eye view. And then there's a little, and then if you separate the parietal and temporal lobes, it's called a horizontal fissure. You peek inside, you can see an internal lobe called the insula. Okay, so we have something called the neocortex in our brain, and in all primates' brains. This is like a brand new, one of the very latest evolutionary innovations. It's really highly complex, and it allows these really sophisticated functions of the brain. It allows it. And it's sometimes hard to assign specific functions to some areas. Like, where is this memory located? Well, we don't know. Where is a thought? Where is this thought particularly? And actually, what is a thought? I have no idea. We don't know. We don't know what a thought is. Okay. Now, there are some areas you can assign specific functions to, and we're going to look at those, particularly sensory areas of the brain and motor areas of the brain. It's the really higher functions that we really can't assign particular areas to, like memory, for example. We don't know where memory is. There is one structure we'll look at called the hippocampus that and we know that that's linked to memory. And that converts short-term memory into long-term memory. But where the memories are located, nobody knows. And remember, and I often talk about this in my other class, non-majors biology class, this old thing about canard about, oh, you only use 10% of our brain. Where did this ever get started? I don't know where to, because that's baloney. You use your whole brain 24/7. Now, you may use some areas that become more active doing a particular task than other areas, but you use your whole brain. I think where that came from is, we usually think of the brain as really sophisticated stuff, figuring things out, rationality, logic, memory, those kind of things. And maybe 10% of your brain is devoted to that. Because we're going to look at, there are huge areas of your brain devoted to the senses, to receiving the senses, analyzing the senses, and telling you what you see, what you hear, what you smell, what you taste. And also motor areas. I mean, even the simplest motor function is incredibly complicated. We're going to get into how many areas of your brain are required to do this smoothly, just to take a drink of a sip of coffee. So huge areas of your brain devoted to receiving and analyzing senses and coordinating muscular movement. Right? Now, and speaking about muscular movement, for example, I'm waving my right hand here. Well, my left brain is controlling my right hand. My right brain is controlling my left hand. And that's called decussation. And that's true for almost everything. There's a few few exceptions. And if the two sides of your body called contralateral. So if I move my right hand, there's a motor cortex in my left brain that is operate those muscles. That's called on the contralateral side. If it stays in the same side, it's called ipsilateral. And there are a few ipsilateral things, but mostly things go contralateral, decussate, and they usually use decussate in the medulla oblongata or in the spinal cord. Okay. Then cerebral lateralization. This means there are differences, physical and functional differences between the left and right cerebral hemispheres. Patalias are physical differences. One pushes a little bit farther forward than the other, all right? And that's pretty minor. Functional is left brain people and right brain people. You know, usually the left brain is the representational. Wait a minute, that's the other one. Usually they think of left brain as logic and rationality, and the right brain as representational, arts and drawing and spatial recognition. So there's a certain amount of differences between the two hemispheres. Okay, the lobes I've already mentioned: frontal, parietal, temporal, occipital, and insula. These are mostly lab things. You have to be able to identify the lobes in lab. And the functional areas, motor and sensory. As I said, huge areas of your cerebrum devoted to sensory and motor functions. Okay, the primary motor cortex, as I mentioned, I am controlling these muscles by the precentral gyrus, and that's also called primary motor cortex in my left brain. Right? And also, I'm talking now because and talking is a motor function, right? I got to contract these muscles in the right way to make these words, and that's called Broca's area, named after a famous anatomist. And the frontal eye field, that's motor function also, right? Where do you point your eyes? That controls your external eye muscles. And then there's a primary somatosensory area, which is in the postcentral gyrus. Oh, gyrus. Remember, your cerebrum is convoluted. It's got gyrus, and so it's ridges. There are ridges and valleys. The ridges are called gyri, or gyrus is one. The valleys are called sulcus, or sulci. And there are distorted representations of your body in the primary motor cortex and in the primary somatosensory area. And somatosensory means touch from all over your body, pressure and touch from all over your body. That's called somatosensory. So it's called a general sense. And there are homunculi there, a sensory homunculus and the postcentral motor homunculus in the precentral. What's a homunculus is a distorted human being, is what a homunculus is. So I'm going to show you what that looks like. I think it's on the next slide. There they are. The primary motor cortex on the left and primary somatosensory cortex on the right. And so your whole body here is represented on your primary somatosensory cortex, the postcentral gyrus. Your whole body is represented. So if something touches you on your left shoulder, a little part of your right somatosensory cortex lights up. Little cells light up there and detect that, and that'll be right around here, shoulder, or trunk, neck, hips, legs, and so you can tell where anything is touching you, and those cells light up. And here's why it's called a homunculus. Look how distorted it is. There's a lot more area devoted to face. Look how much devoted to the face and to hands as compared to arms, trunk, and legs. And why is this? Well, it has to do with sensory fields. And we're going to get to this more in the sensory chapter when we talk about sensory neurons. They detect touch from an area of skin, right? So these called receptive fields, and the receptive fields on your trunk, for example, on your back, are very large, and they're supplied only by one sensory. So one sensory nerve gets information from a quite large area of skin. But on your fingertips and your face, for example, the receptive fields are much smaller. So there's a lot more sensory cells leading from your face and your fingertips than leading from here. And this can be shown by what's called the 2-point discrimination test. So you get 2 points, and you put them a certain distance apart, right? And without the person seeing, you put those on your back, and you ask the person, is that 1 prong or 2? And usually they say it's only 1, even though there are 2. But you do the same thing on your face, and they say, oh, that's 2. Why? Because the 2 prongs are hitting probably the same receptive field and are being relayed in by one particular sensory neuron. It's just getting one stimulation. But on your face, the sensory fields are so small that the 2 prongs are hitting different sensory receptive fields. And therefore, 2 sensory neurons are sending information to your brain. And that tells you that 2 things are touching you, not 1. Okay. So, and then I always bring up how small the area for genitals is. You think that would be a lot more sensitive than that. And there's only a little bit of area devoted to genitals in right in the longitudinal fissure there. Okay. Then we look at the primary motor cortex, and we see that it's almost identical. It's distorted in almost the same way. Lots more of the face and fingers and much less the legs and arms and trunk. But for a different reason. This has to do with how many muscles and how big what's called the motor units are. Now, at the end of the semester, we'll talk about the muscle system, how muscle works. And a motor unit is how many muscles are innervated by the same motor neuron. So on your back, the motor units are huge. There's hundreds of fibers that are controlled by one motor neuron. So they make large, unsophisticated movements. They're not delicate movements. But in your fingertips and in your face, there's lots more, there's small muscles here that control your fingers and muscles of facial expression. There's a lot more motor neurons there. The motor units are much smaller, so they're able to do really fine detail and make all kinds of different facial expressions because of this. And that's why the face and fingers are overrepresented in the motor cortex, like they are in a somatosensory cortex, but for a different reason. Lots more motor neurons that are controlling those, a lot of small muscles being controlled by lots of motor neurons, where on your back, huge muscles controlled by much fewer motor neurons. Okay. Okay, then we talk about sensory areas, the primary sensory areas. That is, that gets the raw sensory information, like raw vision, raw smell, raw taste, raw audition, sounds, and they process it. Okay, and then there's association areas that tell your brain the significance of that. And what is that? Okay, so I see something that's processed, seeing is processed in the primary visual cortex. And then I got to figure out what it is. What is that? And how important is it? And in the primary visual area is the occipital lobe, primary auditory in the temporal, olfactory is in temporal, and gustatory, which is taste, is in the insula. In the association areas, premotor cortex, somatic motor association area. This is also areas where learned skilled motor activities are, like riding a bicycle, playing a violin, are stored in those areas. So the association areas identify the particular sensory input. So you see, so the image of your grandmother is processed by the primary area, and then that sends called association fibers to the visual association area, and that identifies you. Oh, that's grandma. Or auditory association. You process the sound, and then association tells you, oh, it's a Brandenburg concerto. You've heard this before. Olfactory, that's bacon cooking. Or gustatory, that's bacon that I'm tasting. So you get the primary image, primary information is processed, and it's sent to the association area to identify, and also to say how important it is. Right? Okay, functional brain regions, integrating information from different areas. We had an area called Broca's area, motor speech, where you make speech. Well, we also have an area, and these areas are usually in the left hemisphere, called Wernicke's area, that's understanding speech. So I always use to my class, I'm using Broca's area to talk to you, you're using Wernicke's area to figure out what I'm saying. And then there's a Gnostic area. And this is an area where we think of the really higher brain function of figuring things out, logic, rationality, memory, and making sense of information from several areas at once. And it's really a diffuse area in the parietal, occipital, and temporal lobes. Like I said, it's not really located too well. And these are the really higher level areas, the Gnostic areas. Okay, let's call it here, and we'll get back. So this is going to be 203 brain 1, and then I'll catch you on the next video lecture here.